איתך עכשיו.
די זה קשה. Embryonic stem cells have several disadvantages. The first disadvantage is that they may give us, once transplanted in the body, they may form certain types of tumor. And therefore, we must be very careful when using embryonic stem cells not to have the formation of a tumor, so that instead of providing a therapy for a person, we actually creating a new problem. The other problem embryonic stem cell has is that they have ethical problem. This problem is also a regulatory problem. You actually have to destroy human embryos in order to obtain these cells. And the third problem that embryonic stem cells have is that they are very difficult to manipulate in culture. In order to obtain the exact cell type that you would like, we need to interact with many mechanisms. And for some cell types, we know the mechanism by which we can lead embryonic stem cells to the desired cell type, but for other cell types, we don't have and don't yet know what are the mechanisms that lead the differentiation of the stem cell to the exact cell type. Additional problem of embryonic stem cells is the source. Whereas in embryonic stem cells, we have to sacrifice an embryo, a foreign embryo, to save someone else's life, there is a mismatch between the source of the stem cell, which is an embryo taken from another person, and the person that will obtain or that will require the cells. This mismatch presents an immunological problem that eventually will result in the rejection of the new stem cells that we enter to the body and the new cell types that we introduce. In order to prevent that, we will have to require additional technologies like gene modification of embryonic stem cells or human cloning, which also have several ethical problems and regulatory problems. On the other hand, by using adult stem cells, we are avoiding all these disadvantages because adult stem cells are much more safe in regards to forming tumors in vivo after transplantation. And human adult stem cells are taken from the patient itself, and therefore we have no immunological rejection of that stem cell and the cells that it give us eventually at the end. And finally, we believe that obtaining certain adult stem cells, for example, from bone marrow, from blood, and from fat tissues, is much more easier and much more safer and much more easier to manipulate in culture before introducing to the body. By preventing these disadvantages with adult stem cells, we are able to obtain similar results, if not even better results with adult stem cells than by using embryonic stem cells.
There is embryo cells are creating cancer or some cancer. It's more sensitive to be cancerous.
Yes, embryonic stem cells are considered much less safer because they have a tendency to form, in the non-differentiated pattern, they have a tendency to form teratomas, which are large tumor inside the body. And we can actually see that by injecting embryonic stem cells into adult tissues, teratomas are being formed.
So,
in order to avoid that, you have to differentiate embryonic stem cells in culture to a certain mature cell type and using only the mature cell type and not any residues of embryonic stem cell. But this is very difficult to obtain.
So you don't work in it? You don't work?
We do not work with embryonic stem cells, rather we work with adult stem cells.
That's great. Now we'll talk about the partnership of National Children's Leukemia Foundation, the NCLF, and Ariel University. So how do you think it's the match? It's a good partnership and so forth. And
well, I think that NCLF and Ariel University Center were able to form a very successful collaboration. It's a very good matching between an organization that has the will and that has the means to support research and to support specific research with cancer and in very, very attractive avenues and very novel avenues of applying stem cell technologies for cancer therapy on the one hand, and the Ariel University Center, which provides the facilities, which provides the know-how, which provides personnel, students that are highly qualified in the field of molecular biology and in the field of stem cell research. And by sharing all these advantages together, I think that the collaboration is able to support this research and eventually may in the long term lead to a very successful